Okay, welcome everybody to this third CCP5 seminar, third seminar in this CCP5 series on looking at the software that's in use in the UK simulation community and what people are doing with it and what functionality is out there. When we were setting up this seminar series, we were very keen to get somebody from the soft matter simulation community, people who do molecular simulations of not just minerals and hard crystals, but also of soft matter. And we couldn't think of anybody more appropriate than Richard Graham at Nottingham, who will be our speaker today. So for those of you who don't know Richard, Richard is a professor of applied maths at the School of Mathematical Sciences in Nottingham. He did his PhD at Leeds with Tom McLeish and Oliver Harlan in 2002. And he has done postdoctoral work in Michigan and Leeds before moving to Nottingham in 2007. Richard works on multiscale modeling for molecular systems, spanning chemistry, molecular simulation, coarse graining, and continuum work. And he's particularly interested in linking together those different levels of modeling. And Richard works on polymer dynamics and crystallization, a subject close to my heart, along with thermophysical properties of impure carbon dioxide. So the format for the seminar will be as per previous seminars. If people do wish to ask questions, then please click the raise hand button in Zoom if you have one, or type a question into the Q&A and the chat. I think if you do raise your hand, all I can do is invite you to type your question into the chat anyway. So that's probably the default starting option. And there'll be some time for questions at the end as well as during the talk if people want to raise questions as we go along. So without any further waffle, I will hand over to Richard and invite you to share your screen, Richard. And the floor is yours.
Okay, thanks, David, for that introduction, and thanks for the opportunity to present. I'm really looking forward to talking about this work to you guys, and thanks everyone for coming. So the brief is to talk about the software for molecular dynamics that I've been using and what I use it for. So I'm going to start by making a confession, which is that this project was the first project that I'd done where I used molecular dynamics. So that really positions me as a user of the software. Rather than an innovator. So we weren't adding new features to the software, and actually I was interested in what we can learn from molecular dynamics to help with the higher levels of modeling. I was very lucky to recruit as a postdoc for this project Mohammed Anwar, who had previously done some work on the software side of things, so was a real expert in molecular dynamics and knew about how to add new features to the software, and had in fact added himself some of the features that we were then relying on for this project. So I made Anwar a co-author on this presentation, because essentially all of the molecular dynamics results were as a result of Anwar's calculations. Okay, so another thing is that this is it was a large multi-scale project. We had a lot of collaborators. Some of them were people directly involved in the project as co-investigators. So these are the ones listed at the top. Some of them were previous group members of mine who have contributed stuff that we relied on for this project. And then other people such as Zue Wang and Jorge Ramirez, who are essentially old friends of mine who really helped me with some of the technical side of the project. We also had some industrial collaborators with Dow, SCG, and who are both chemical companies, and Autodesk, who are a computational fluid dynamics company. Okay, so I'm going to spend a little bit of time introducing the area and bringing in some of the features that are going to inform sort of how we ran the project and how we designed the molecular dynamics simulations that we did. So I'm going to contrast here crystallization of small molecules, essentially small, simple molecules, how they crystallize in a very simplified form. Essentially, they're small enough that they can interlink and form structure, compared to polymers, which are long molecules that still like to crystallize, but because they're long chains, they tend not to crystallize as an entire chain at once, but subsections of the chain crystallize, and that creates these sort of dangling ends here of the non-crystallized part of the polymer, and that has really rich and important consequences for how the polymers crystallize. They have this sort of local structure, then they form these sort of sheet-like structures where these loops that you can see here are the dangling ends. Those sheets then form layers of sheets and ultimately grow to form these sort of spherolytic structures. So there's something kind of really interesting going on, which is the start of the crystallization happens down at this length scale here, but the point at which they're detected by experiments is kind of up here. So there's this gulf between where the key initial processes are happening and then the point at which the experiments start to see the result of that process. There's another thing that I think is really interesting and particular to polymers, which is related to their long chain nature, which is what happens when you put these things under flow. And this is completely ubiquitous in polymer processing. Anytime you want to process a polymer, you have to flow it. And that's generally going to bring about some crystallization. So here are some sort of prototype experiments to show the effect. We've got a polyethylene polymer flowing through here, and it just expands out into this wider region. So it's a sort of prototype die for some simple polymer processing. This is the start of the problem. Then as the crystallization, as the flow carries on, we start to see the crystals appear as these darker regions here. And you can see that they're preferentially forming in specific regions, particularly along the edges of the slot and in this exit region here. And this is essentially because wherever the flow is strongest, wherever there are the highest flow rates, the strongest stresses developing, that encourages crystallization to happen. So then as the flow progresses, we can see the crystallization is happening again in these regions of strong flow. And this is a key idea in polymer processing because it gives a way of controlling where the crystallization happens and therefore essentially controlling the final product. And the types of variables that processors use to control this are the temperature, the flow rate, and the molecular weight of the material that you're processing. They generally don't like to change their equipment, but they can change what they put in, how hot it is, and how quickly it goes. So the central idea around flow-induced crystallization is that somehow the flow helps nucleate the initial crystals, and that templates the growth for the kind of crystallization you were seeing in the previous slide. So we're just going to have a quick look at what nucleation is because it's going to be a central idea in this project. So the sort of classical view of nucleation is that if we imagine that we've got a liquid, which is supercooled, so it's below its crystallization temperature, that means that crystals want to form because bulk crystals have lower free energy, but there's an interface between the crystal and the liquid that has a cost. So you get a free energy gain proportional to the volume of the material and a cost proportional to the surface. And it creates this characteristic barrier where the surface energy cost dominates for small nuclei, and ultimately the bulk energy wins if you make a big enough crystal. So we have this region where little crystals are unstable. So in order to create a crystal, the nucleus has to reach this critical size here. And to do that, it has to diffuse over this unfavorable barrier. And that's very difficult on a molecular scale. You have to have a sequence of unlikely events happen in order to get over here. And then once you get over, you can grow spontaneously. And a little bit of analytic theory will show that the time to nucleation is exponential in the barrier height. Sorry if you can hear my daughters in the background. All hell is breaking loose. I apologize for that. Okay, and so the barrier height is here, and so we get something that's exponential in the barrier height. So the nucleation rate is very sensitive to how high this barrier is. So now we can ask the question, well, how does flow help nucleation? And we know that polymers unravel under flow. Here's a visualization of some DNA molecules. And so the idea is that as you flow the polymer, it unravels, and that takes it closer to the structure that it needs to be in order to crystallize. And so that lowers the nucleation barrier. So here's a kind of cartoon of what we believe is happening. You stretch the polymers out. While the polymers are stretched, nucleation barrier is much lower, and that encourages much more rapid nucleation, which is why we see more crystals in the regions where the flow is strong. So the job, and it's been a job for a long time, is to attach some numbers to this cartoon. Okay, but before I dive into sort of how to attack this from a modeling point of view, I want to talk about some experimental difficulties as well. So this is a fairly long-standing problem, and I want to explain a little bit about why the experiments are difficult and in some sense less than ideal. Okay, so this is actually a simulation of some nucleation in polymers under flow, but I really want you to imagine this from an experimental point of view. So what I'm saying here is that this is a simulation of nucleation as it happens. The colored bits are little bits of crystal that we're detecting in our simulation. And the key event happens on an extraordinarily short timescale and on a very short length scale as well. So this thing is now nucleated and is growing spontaneously. So that important event has happened in perhaps a few nanoseconds and on a very short length scale. And it makes it extraordinarily difficult to view in experiments. So the key thing is happening on a time and length scale that's much quicker and much finer than can really be detected by experiments. There's a second difficulty, which is that real polymers, particularly crystallizable ones, are almost always polydisperse. So from a modeling point of view, what we'd like is our molecular weight distribution to be a lovely spike like this, which says that there's essentially just one molecular weight in the system. But then reality kicks in and real crystallizable polymers are pretty much always polydisperse. And so we've got a range of molecular weights. And this is true even in the best, most controlled experiments. And so what that means is you've got some short chains that are in the low molecular weight tail that are very hard to deform. The flow just doesn't catch hold of them because they're so quick. And at the other end, you've got these really long chains that are very easily deformed because they're so slow moving. And so what this means is any crystallization experiment has a kind of combined effect of these very deformed chains and these very weakly deformed chains. And the things that are measured are essentially a combination of these different species of chains. And this makes it very, very hard to get at the key physics of the problem because you can't do an ideal experiment. You have this mixture of molecular weights. And even if you can, the key step is happening on such a fine time and length scale that you can't really see it. You can just see what's happening afterwards. And so this suggests that molecular dynamics could be a good way to step into this gap, a sort of complement to experiments. The idea being that we've got the spatial and time-based resolution to resolve nucleation. And of course, it's easy to dial in monodisperse chains into a simulation. So there's this idea that perhaps we can complement experiments by using molecular dynamics. Okay, but of course that brings difficulties of its own. So perhaps some of the things that experiments are strong at are a real problem for simulation. So one of the things we can start to ask about is how rare is nucleation? So nucleation, I think, has this kind of curious feature in that when it happens, it's very fast. It happens on a really short time scale, but it's phenomenally rare, which means if you sit around and just wait for it to happen in a simulation, it generally doesn't happen. So we should think about nucleation as being slow in the sense that it takes time to see a nucleation event, but when it happens, it's virtually instantaneous. It's like that. So to give a feel for how rare nucleation is, I'd like you to imagine staring into a glass of beer and seeing the bubbles appear. So each of these bubbles is the effect of a single nucleation event. And we can do a very hand-wavy back of the envelope calculation of just how many attempts at nucleation are going on in this glass of beer. And so we could say, well, maybe there's Avogadro's number of particles in this system, and so let's say 10 to the 23, and they're attempting to nucleate on some microscopically fast timescale. Maybe every second each particle attempts to nucleate maybe 10 to the 7, 10 to the 9 times. And so we've got this unimaginably vast number of attempts at nucleating. But when we look at the system from a bulk, what we see are a countable number of nucleation events. With enough patience, I could sit down and count the number of bubbles, and we have a few hundred every second. So it's unimaginably rare, the nucleation. And of course, that's a massive problem for simulations, because we can't possibly simulate a system that's anywhere near this big. And even if we could, we would only see a relatively small number of nucleation events. Okay, so nucleation in polymers presents two specific and quite different computational difficulties. So the first difficulty is that polymers are slow. We're interested in big, long polymers. They're large molecules. It takes them a long time to move and to relax. So we have this sort of classic problem of slow diffusion, which I'm sort of trying to show here in this movie. And in some sense, that's analogous to trying to complete a really large jigsaw. You need to give me a little bit of artistic license on here. It's not an exact analogy, but you should imagine that you're building the jigsaw, and every now and then you accidentally take a piece away. So it's not a complete backwards and forwards process. But what I want you to get out of this is the idea that diffusion in polymers is rather like a series of a large number of small steps, and you can monitor progress. You can see, oh, well, I'm half finished. Okay, now I'm 3/4 finished. You can see the answer coming. So the process is many small steps that all build up together to create the result that you're looking for, which is the relaxation of the polymer. Nucleation, by contrast, is very different to that. It's fast but rare. So I think it's analogous to trying to flip 10 coins in a row. And the analogy is helpful because there's a small possibility that it could happen the next 10 flips, could be what you need for success. But the overwhelming, so when it works, it's quick, but the overwhelming probability is that it's not going to happen. And in fact, you'll make no progress, and there will be frequent failures. Maybe you flip 3 heads in a row, you get a tail, you start again. Maybe next time you get 5, you start again. So it's rare and characterized by frequent failures. So you need to be aware of these two different difficulties in terms of simulation. They're both a problem for molecular dynamics for nucleation in polymers. Okay, so to attach some numbers to these sort of time and length scales, I've got a plot here of the time scale in seconds, and you could argue that virtually this entire range is relevant to polymer crystallization. And we've got a plot of molecular weight down here. So if we put some numbers in here, we can see that a successful nucleation event is very fast. So I'm thinking about the time that the nucleus first appears to when it successfully nucleates. So by analogy, the time at which you first flip a 10, that then go, first flip ahead and go on to flip the remaining 9 to get the successful event. That event is very short, but the time typically to be able to detect the crystallization in experiments is sort of much more up here, perhaps maybe a fraction of a second if you've got some really nice experiments, but can be much longer. And now we can ask, well, where do the crystallization experiments happen? And they happen typically in this range of timescale, so a few tenths of a second up to minutes or hours, depending on how patient your experimentalist is. And what sort of molecular weights do we look at? Maybe the lowest is perhaps 1,000 monomer units up to approaching 1 million. So this would be, processors would say, well, this is a very low molecular weight material down here with just 1,000 carbons in your polyethylene. Up here is ultra high molecular weight. So now we can ask, well, where do molecular dynamics simulations sit in here? What's feasible? And the answer is that molecular dynamics sits down here. It's only possible to do relatively modest chains because you have to fit them in the box. And we can only simulate a relatively short period of time, but we can do that to, of course, a very high spatial and temporal resolution. So, maybe we can argue about the edges of these, boxes. But the point is that this gap here between, particularly in time scale, between the simulations and the experiments is real. We can argue over exactly how big it is, but there's this separation. So this creates a difficulty in this idea of using simulations to complement experiments. I mean, clearly there's some complementarity because the molecular dynamics operate in a region where we are interested and we have good time resolution there, but they're going to struggle to access the long time scales and high molecular weights that are really relevant to polymer processing. David, I've just noticed there are a few questions in the chat. Do you want me to take a look at those in situ, or can we wait till the end?
I was going to wait for an opportune moment to jump in and ask you exactly that question. So it's up to you. If you'd like to take them now, then that's great, or we can save them until the end.
Yeah, let's take them now. Yeah.
So can you actually see the questions, or do you need me to?
I think it would. I'm not sure I can.
Okay, so I'll pretend to be the person who's asking them. So the first question was about the nucleation for unraveled polymers and whether the nucleation is driven by van der Waals interactions or by collisional processes.
That's a good question. It depends on the polymer. For most typical processing polymers, it's driven by van der Waals interactions ultimately, but excluded volume is very important as well. So it depends whether you think of those as part of the van der Waals interaction. There's an attraction that joins them together, but also the excluded volume is important. And that the reason why excluded volume is important is that segments that are lined up in the same direction find it easier to sit next to each other. So it's a sort of combination of the two. I'll be showing a force field that puts in all the interactions that we need to describe the crystallization a little bit later on.
So the second question was about the flow-induced crystallization. What is generating the flow typically? Okay,
so that depends. There are really two situations. So one of them in polymer processing, it's generally a pressure-driven flow. So it's something like you're trying to make the polymer flow through a channel, and so you give it a big push from the other side. So there's a pressure that drives the flow. Another classic one is film blowing, where essentially they blow a very, very large bubble of polymer film. That's how they make plastic bags. And so again, that's pressure that's blowing up this balloon that creates the flow. In a lab situation, it's more typically done in a rheometer, which is a machine to impose a well-controlled flow, typically shear. So a sort of idealized picture of how they do that, if you can see me on the video, is you have two plates and you hold one plate fixed, then you just use a force to drive the plate that way to impose the flow, shear flow. Mostly the shear rheometers do it in a circular arrangement so that you can, so you don't run out of space. So there you're using a stress to drive the top plate, and then the internal structure of the fluid spreads that flow through between the plates.
And then the third question, I think, was inspired by your beer video, which was in that video. It looked like there were preferential places on the side of the glass where the bubbles were nucleating. So I think the question is, what are those preferential centers, both in the context of the beer and in the context of the polymer crystallization? Are they sort of nucleation seeds that promote the polymer crystallization?
Okay, yes. So when someone asks me a question along this line, I wonder whether it's because they've spent a lot of time staring into the bottom of a beer glass, watching the bubbles appear. The other possibility is maybe it's someone that's involved in your crystallization in the real world project.
It is not, I can tell you.
Okay, but it's a good question. Okay, so what's happening is this is the effect that's known as heterogeneous nucleation. And so what it means is that there are, anywhere where there's an imperfection, there's a reasonable chance that nucleation is more favored there. So what tends to happen with beer glasses is that any little nick in the glass, or even the glass itself, will preferentially create nuclei. So if you look really closely, you'll see specific points on the side of the glass that's creating a stream of bubbles. And that's because the nucleation is a bit easier at these sites of imperfection, generally because the surface energy is a bit lower. And so that tends to dominate the nucleation in something like a beer glass. What's the effect on polymer crystallization? So that's interesting once you add flow. So what generally tends to happen is that if you don't have flow, the nucleation is dominated by imperfections in the polymer, so bits of dust, little bits of catalyst that have been left in the polymer. And you can see this. The way that you see it is you crystallize the material without flow, and you note where it crystallizes, then you melt it again and crystallize it again, and it crystallizes in exactly the same places because that's where the imperfections are. What's interesting with flow-induced crystallization is that once you add flow, you make the bulk crystallization sufficiently favorable that actually that then starts to dominate. So when you do the similar experiment in a flow-induced case, you can see that the distribution of crystals that you see is different each time you repeat the experiment. So you're not seeing this heterogeneous nucleation. So the way it tends to happen when you're comparing with experiments is people typically assume that there's a sort of background amount of heterogeneous nucleation that appears the second that you cool the material down. And then the additional nuclei that you see during flow are the result of genuinely random bulk homogeneous nucleation. That's generally how it's tackled. Of course, it's another nice feature of molecular dynamics is we don't have any catalysts or other imperfections floating around in the material, so we're genuinely sampling homogeneous nucleation.
Okay, great. Thanks, Richard. I'll let you move on, and I'll jump in if another build-up of questions occurs.
Okay, so we were talking about this gap between molecular dynamics simulations and crystallization experiments because we'd like to use molecular dynamics to complement the crystallization experiments. So a classic way of approaching this problem is to use multiscale modeling. You've got a really wide separation of time and length scales, so you're going to use different theoretical techniques that are relevant on different length scales. So this is a scheme that we came up with. So we're working down at the level of molecular dynamics simulations. We're then going to add to that a kinetic Monte Carlo model that's got a sort of limited resolution of a discretized nucleus. I'm going to talk about this in more detail later on, but it's a sort of minimal generalization of classical nucleation theory, but it's still a simulation. Then we're going to go to continuum level models, which are essentially deterministic differential equations. So we're going to try and get all the stochastics out of the problem and solve the problem via differential equations. And the reason that we want to do that is that then we have something that can be put into a finite element calculation and is suitable for modeling polymer processing. So this, of course, idea of multiscale modeling is common across many, many areas of science. And this is just a sort of specific scheme that, that we've worked with. There's a couple of reasons why we set the scheme up like this. So one of them was so that we could partition out the physical approximation. So every time you move between a different level of modeling, you're often making both physical and mathematical approximations. But with this scheme, we could separate the two out. So we made some quite drastic physical approximations going from molecular dynamics, where essentially we resolve everything in detail, to this much more limited discretized model. But because we're solving it via a kinetic Monte Carlo algorithm, the mathematics of our solution of that model is rigorous. We're producing a rigorous statistical solution to that model. So any problems that we encounter, we know are down to physical approximations. Then when we make the step from kinetic Monte Carlo to the continuum scale model, we're then introducing mathematical approximations. So if we have problems at this point, we know that it's because of the mathematical approximations that we've set out. So we arranged things so we could separate out those two processes. Another thing that we did was we worked very hard at the interfaces. Most of the effort of the project, aside from doing the molecular dynamics simulations, was working at the interfaces to these problems, how to get the techniques to talk to each other. And then a final nice feature that we found from this setup is that then when we've got a working continuum scale model, we can start using that to ask questions of our molecular dynamics simulations. So we'll start to generate predictions at these sort of macro scale length scales from our model, because we can run it on more interesting systems. And we can then use those to inform our molecular dynamics simulations. So interesting things that arise out of these continuum models, or questions that we can't answer, we can then go back to the molecular dynamics and design simulations to help us deal with those problems. Okay, so now I'm going to talk about our molecular dynamics simulations, and because of the sort of brief and the audience, I'm going to spend perhaps a bit more time talking about design of the project, what software we had, what clusters we had access to. And I guess the main reason we're using molecular dynamics is we can have monodisperse polymers and we can see everything that's going on. Okay, so what hardware and software did we have access to? So we use predominantly the Midland Plus supercomputer, which is a tier 2 regional cluster, which is hosted at the University of Nottingham. So it's considerably bigger than the Nottingham in-house HPC, but is smaller than, say, Archer, which is a national facility. So there's some information about the sort of technical side of this cluster. We could run each node had 28 cores, but we were running multi-node simulations. We had to apply for time on this cluster, and we typically attained about 300 to 500 CPU hours per quarter, and that was over a period of 3 years. So we got significantly more compute time on this machine than we would have been able to get on the Nottingham HPC. So I just should mention that David Quigley, who's the chair of this session, was one of the lead investigators on this project. So we were really grateful to have access to this resource. And also a really nice result of using this cluster was, because the way the project was set up, we had to rationalize what we were going to do in advance. We had to show that we had good scaling on this cluster. We also gained from the research software engineer expertise on this cluster. So it really helped us with the planning of the project. I think in a way, because this was my first project, having these constraints of having to justify what I was going to do, how much it was going to cost, was really, really helpful, and it's something that I'm going to be using in future molecular dynamics projects. Okay, in terms of software, what did we have? So we began by using the Espresso software, which is a molecular dynamics package designed for soft matter. And we were doing this because my postdoc, Mohammed Anwar, had expertise in this software, and he'd actually done some of the modifications that we needed. We also worked with LAMMPS, which is probably a better known piece of molecular dynamics software. And what we found was that in some cases, LAMMPS was up to 5 times faster than Espresso. So as the project went on, we kind of migrated across from Espresso onto LAMMPS. We didn't really have time to explain why LAMMPS was so much faster. The most obvious reason is the Midland Plus cluster had a significant amount of research software engineer time getting the compile right for LAMMPS, customizing the compile for that particular architecture in a way that there wasn't for Espresso, because we were the only Espresso users on the cluster. So I think there's a take-home message there. If you're doing these massively parallel calculations, it's really, really worth comparing different types of software and bringing in some experts on getting the compile and the setup right for that software, because it made a big difference for us. Okay, so I'm going to start to talk now in a little bit more detail about the simulations. So we were simulating polyethylene, which is probably the simplest crystallizable polymer, and it's very widely used in processing. And it crystallizes into this shape here, where the chains lie in what's called a planar zigzag, and then the zigzags sit next to each other to create these favorable van der Waals interactions. And we used a force field that was designed with this crystal structure in mind. It's one from the literature, we didn't develop it. And essentially the van der Waals interactions are taken into account by Lennard-Jones interactions for every bead in the system. The bonds for the polymer were modeled as a harmonic spring. We introduced a bond angle potential, so there was a cost to bending the bonds in this direction, and a dihedral potential, which is between four of the atoms. And this force field was designed essentially to recreate this crystallization structure, and a lot of work went into parameterizing this force field properly prior to our project. It's a fairly well-established force field. The other thing that we introduced was an order parameter, which was a way of detecting crystallization in our simulations. And it's actually a fairly simple idea. Is it just that our order parameter just looks for clusters of similarly aligned chain segments. So it looks for the alignment of chain segments, and then looks for clusters of segments that are spatially close and aligned in the same direction, and assigns those as crystalline regions. Okay, so I'm going to talk a little bit more about the detail of the simulation and some of the things we were weighing up as we designed the simulation. So we were starting to ask, really, what's the biggest molecular weight that we can feasibly do within the constraints of the computing facilities we've got access to. And we needed to answer two questions. Can we fit the polymer in the box? And that's what this first calculation is. And essentially the constraint is that the box needs to be big enough to fit the chain in. So there's a scaling theory that tells you how the size of the chain, the radius of gyration, scales with the molecular weight. We needed a box that was big enough to fit in that chain. And so then that told us how our how our number of atoms scaled with the molecular weight, and we knew that the time step was going to be proportional to that because we're using a finite length cutoff. So that gave us an estimate for the scaling of the time step cost with molecular weight. And then the second question is, can we relax the material? So we set this thing up in an arbitrary configuration. We need to give it some time to fully forget that arbitrary configuration. And for polymers, that's very, very, very long. So there's a well-known result from entangled polymers supported by theory and experiments that the time to equilibrate scales as the 3.4 power to the molecular weight. So you bring those two together and we've got a scaling of almost the mass to the 5, the total cost of the simulation. So we needed to get the molecular weight right. A little too high and we would have pushed it into a region where it's not feasible. Too short and we weren't really pushing the boundaries of what sort of molecular weight we could run. So we came up with a molecular weight of 1,000 carbons, and we estimated the equilibration time for that by looking at a series of shorter polymers using this scaling. And we also then, so once we'd set up that simulation for 1,000 polymers, we felt that that was achievable. We then calculated the cost per time step, looking at different number of nodes, looking at different clusters. And from that, we could estimate the equilibration time in days. So if we were using one node on the University of Nottingham HPC, it would have taken 18 months to fully relax the system. So that's essentially 28 processors on a single node. Quite a bit faster on Mid Plus, again, probably because of the better compile. But if we move up to 10 nodes, then we get a significant drop, and now we're into the region of months to relax this system rather than years. So we've got an achievable drop off, and we tried slightly larger, and we didn't get enough of a return that we felt it was worth going to more nodes. When we were doing shear flow, we imposed this shear flow via periodic boundary conditions. There's an established technique to do this, which I don't have time to discuss in full detail. Okay, so now we've decided on our size, we've got our access to the cluster. We need to do a sequence of runs in order to generate our data. So the first thing we need to do is fully relax the system. So we had an estimate for how long that would take. And what we're trying to get out of this is a series of fully relaxed configurations so that we're ready to do meaningful simulations with those relaxed configurations. And so you can see that the amount of time that this took was about a little more than 1/4. So about 3 months of simulation time just to relax these materials. Then we need to work out some of the relaxation times. So we did that via a nonlinear shear simulation because new relaxation times appear when you impose shear. So this was a much shorter simulation. We only needed to do it once, and that would tell us the chain relaxation times. We needed to know how those relaxation times would change with temperature because we wanted to run at different temperatures. So we did a series of runs at different temperatures, which I'll talk about in a moment. These were extremely short runs, but we needed to do a reasonable number just to scan out the temperature space. And from that gave us the dependence of the relaxation times on temperature. And then finally, we were ready to do our actual shear and crystallization runs from which we'd get our nucleation data. So now these runs are somewhat short compared to our equilibration runs and even our nonlinear shear runs, but we want to do a large number of them because we want to span out different flow rates and different temperature. And these will actually provide us with our nucleation data. Okay. So this is how we got the Rouse time from the simulation. So the Rouse time is the time associated with stretching of the chain, and it was believed that this is the most relevant time scale to the to flow-induced crystallization. And so essentially we ran two runs, one where the system remains in remains essentially in linear response, and that allowed us to extract a modulus. So this is stress against time, and this region here gives us the modulus. And then we ran a slightly higher rate where we saw this overshoot, and we used a model that described this overshoot to extract a relaxation time associated with that overshoot at one temperature. We then wanted to work out the dependence with temperature. So essentially what we did was a series of linear stress relaxation. So this is stress against time for a series of temperatures. And then there's a well-known concept in polymer physics, which is time temperature superposition, which says essentially when you change temperature, you speed up or slow down all processes. So it's just a question of sliding these different relaxation curves onto each other to find out what's the shift that you get when you change temperature. So by what factor do the timescales change when you change the temperature? So these were really short runs, but really important because they helped us to predict how the relevant timescales change with temperature. Okay, so now we're ready to run our actual production calculations. So here we plotted the shear stress against time. So again, it sort of generally goes through this overshoot, but we're now below the crystallization temperature, so we can see nucleation events as they happen. So we've built up this histogram of nucleation events. So we can see that if we flow at a particular rate, we generally see nucleation in this region here. So we get a measure of the average nucleation time and the distribution of that nucleation time. We can repeat that at different flow rates and then repeat at different temperatures. So this is sort of spanning the space, seeing when this flow-induced nucleation happens. And then we can pull these things together and say, well, what's the average time to see that we first see a stable nucleus against, this is our flow rate down here, 4 different temperatures. So we were sort of starting to map out the parameter space for this system. And so at this point, we were just starting to think about different ways to analyze the data. And at the same time, a rival group published some really, really interesting simulations, which actually were extremely useful to us. So they looked at a much shorter system than us, just 200 carbons, but that meant that they could do many more simulations looking at different types of flow, and they produced this really, really important result. They were able to collapse their nucleation rate onto an order parameter that they could extract from their simulations. So essentially the order parameter they used is this pneumatic order. So it's a well-known order parameter in polymers. Essentially what you do is you pick the principal direction of orientation and you look at the angle that each bond makes with that, and then produce the average of that through the Legendre polynomial. And this gives you an order parameter, which is 0 when the whole thing is completely random, and goes to 1 when everything is when everything is completely aligned. And they showed that this quantity was the best at collapsing their simulation data. So we took this idea and we checked that our simulations were consistent with this idea, and they were. So we took this as one of the key modeling messages, that this order parameter is the relevant one that you should use for how flow affects nucleation. Okay, so that's the first idea that we took from our molecular dynamics simulations, and then we started to try to translate that up the length scales into our different models. So I'm not going to have enough time to talk about these models in detail, but they're a really important part of what we achieved, and they're really setting the stage for what simulations we should be doing. So I want to give you enough information so that you can understand that. So we started with a kinetic Monte Carlo model that I developed a while ago, and so this model is, the idea is it's a simple generalization of classical nucleation theory to polymers. So it imagines that the nucleus is built up via 2 steps: adding new stems, like here, and of course the corresponding move of taking stems away. And the second type is taking an existing stem and lengthening it, and also shortening it. So it's this sort of very simplified picture of the physics. So we've introduced new physical approximations, but we solve this rigorously stochastically via a kinetic Monte Carlo algorithm. So it's still a simulation, but a much, much simpler and cheaper simulation. And what's nice about this model is it now allows us to introduce different types of chain, so different chain lengths, different degrees of stretching, and we can look at how those cooperate to perform these moves. And we know from molecular dynamics that the key order parameter that we should be using to measure how flow is changing the move rate is this pneumatic order parameter. So we took that idea and embedded it into this model, but now this model can run multiple chain lengths. And a really important feature comes out of this model. Once you write these rules down and start solving this model, it becomes apparent very quickly that the concentration of long chains in the nucleus is much higher than long chains in the melt. And it's because the long chains stretch more, they attach faster, and so your nucleus contains more long chains than you would imagine if you just thought that the concentration should be the same as in the melt. So this model predicts that long chains concentrate in the nucleus, and this is actually a fairly controversial idea, and I guess still is, because there are some quite famous experiments on well-developed crystals saying that it's not true. But what we're predicting is something a little bit different, which is that in the early stages they're highly concentrated. Our model actually predicts that if you have well-developed crystals, eventually you have to get back to the melt concentration. So anyway, so we've got that kinetic Monte Carlo algorithm. We now want to take the stochastics out by introducing mathematical approximations to solve this system analytically. So the way that we did that was try to project the model onto a single dimension. And the single dimension that we used was the size of the nucleus. So we're now taking another step towards classical nucleation theory, but we want to write down a model that's consistent with our kinetic Monte Carlo algorithm. So essentially what we're doing is saying, well, here's all the ways of making a nucleus size 1. Here's all the ways of making a nucleus of size 2. Here's all the ways you can make a nucleus of size 3. And we want to group, and the model tells us the rules for skipping between these states, but we'd like to group these states together to lower the dimension of the system, to get it down to 1 dimension. And the task becomes working out what are the transition rates between these sort of clumped states. So it turns out, with a little bit of thinking and a bit of statistical mechanics, that this is a solvable problem. So the way that we did it was to work out what the free energy barrier is, and I'll show that calculation in a moment, and then to work out what the kinetics are for this projected system. Now, the kinetics were pretty straightforward. We could just observe those directly from our simulations by watching it skip between these states. And the free energy barrier required an analytic theory, which I'll explain in a moment. But the point is, if you can get these two quantities, then you can do a sort of classic Kramer's problem. So a one-dimensional crossing of a barrier, which is a problem I think that was solved in the 20s. So there's some well-established mathematics around that problem, but the task is to get down to a low enough number of dimensions that we can actually do the calculation. Okay, so this is how we calculated the nucleation barrier. Now, bear in mind, this is for a large number of different species, all with different degrees of stretch, so different attachment rates, and we want to write down a model that's consistent with the kinetic Monte Carlo algorithm that I set up. So it turns out, okay, so we've got many different species with different values of this order parameter and different concentrations in the melt, all trying to attach to the nucleus. And so we want to write down a free energy of a nucleus of this size. So we set up a few variables. I'm not wanting you necessarily to understand the mathematics in details, but just the sort of flavor of how we set up this model. And so from these variables, we then wrote down a nucleus free energy. So accounting for things like entropy change from the melt to the nucleus, entropy within the nucleus, so different ways of arranging stems and monomers inside the nucleus. We've got a sum over the different species. So these are the different molecular weights. We've got a term that's this order parameter that we got from molecular dynamics. And then we've got some terms here that look like classical nucleation theory. We've got a bulk gain and a surface cost. And then we want to minimize this free energy with respect to these constraints. So this constraint just says that the total, these are the fractions of stems of a particular species and monomers of a particular species. So both of those have to add up to 1. So this constraint just normalizes the system and just says that your fraction of monomers has to add up to 1 and your fraction of species has to add up to 1. So we can minimize with respect to this constraint, and then we can produce a model that is almost analytic. It's not quite analytic because to do this constrained minimization, we need to solve a one-dimensional nonlinear route. So it is numerical, but it's very, very cheap. A one-dimensional route finding is extremely easy, it's extremely cheap to do. And then everything else works analytically. And now we can predict our nucleation barriers that we get from our kinetic Monte Carlo simulations. So we now have a handle on how drastic are our mathematical approximations that we've introduced in here. And the answer is not very drastic at all, except perhaps a little bit in very extreme circumstances. Okay, so now we've got our analytical model that's come up through the length scales. So the ideas have been germinated in molecular dynamics. These key questions came from that. We then introduced physical approximations to get to our kinetic Monte Carlo. And then we were able to solve that kinetic Monte Carlo algorithm nearly analytically. So we've got a tool that we can now use to do fast calculations with. And that lets us compare to experiments, because we can do high molecular weights, we can do these very long crystallization times. And because the model's cheap, we can play around with the parameters. We can explore the parameter space and see whether there are plausible sets of parameters that describe the experiments. You can't do that with simulations because you can't run multiple parameter values within your simulations because they're too expensive. Okay, so here we did a comparison of the measured value of the nucleation rate against shear rate. So this is just a polymer that's shear is imposed on it, and they watch the nucleation nuclei appear. They see them, when they grow into bulk crystals, but they know each bulk crystal has corresponded to a nucleation event. So you literally just count the nuclei as they appear, and that gives you a nucleation rate. Then you change the shear rates, look at different flow rates, count the nucleation rate again, and then look at different temperatures. So the points are the experiments, and the lines are different levels of theory. So this dotted line here is a sort of naive generalization of the order parameter. So this is sidestepping the kinetic Monte Carlo step and just saying, I'll average the order parameter over my different molecular weights and assume that that controls nucleation. And then that gives essentially exponential behavior. So this is a linear against log plot. So straight lines are exponentials. And it gives this exponential behavior that's okay at low rates, but there's a clear curvature in the experiments that if you miss out the kinetic Monte Carlo step, the model doesn't capture that. But if you do that kinetic Monte Carlo step, so use this free energy that we wrote down, and in particular account for the fact that long chains are more concentrated in nuclei than they are in the melt, then you will pick up this curvature and get much closer to the experiments. So we made a couple of conclusions from this. The first one is this curvature in this log linear plot is an experimental signature of a long chain enhancement, which is there in our model. So that's something that emerges from the physics of polydispersity. The other thing that we noticed is, going the wrong way, that the model gets a bit too excited and predicts probably too much long chain enhancement once you're out at really high rates. So it captures very nicely the early upturn, but there's some evidence in the experiments of a saturation, and the model is saying the exact opposite, that essentially the concentration gets stronger and stronger. So we did a little bit of thinking about that and proposed some slightly different physics that alleviate some of this over prediction. That helps a bit, doesn't remove it completely. And so this is new physics that we've not yet tested against our molecular dynamics, but does seem to help. Okay, but now we've reached the point where we can use our model and the predictions that it's making. So our model is predicting long chain enhancement is really important to describe the experiments, but the experiments can't tell us whether that's true or not. We can't see a critical nucleus in experiments. We can't measure its concentration. So at the moment, the only experimental evidence is via this curvature. But we're now in a position to go back to the molecular dynamics and ask that question directly, because we can see the nuclei as they appear. Can we see long chain enhancements in our molecular dynamics simulations? Okay, so we're in a position to do this. But the first thing we have to remember is that to see long chain enhancement, we need at least 2 different molecular weights. If we run everything with a single molecular weight, there's only one species. that can't be enhanced in the nucleus because all nuclei contain only that species. So we go back to our material that we were looking at, which is this C1000 at 100% concentration, and we want to make some blends. But what we don't want to have to do is re-equilibrate the whole system. Remember, we spent 3 months equilibrating this system, so it would be nice to be able to use these fully relaxed systems to help generate some blends. So the way that we did that, I mean, in some sense, it's rather small-minded. We just went through and we cut 50% of the chains in the middle to create a mixture of 50-50 by volume of 1,000 chains, 1,000 carbon chains, and 500 carbon chains. Now, this system's not fully relaxed, but it's close to fully relaxed. So then we equilibrated it, and the way we did that was essentially run a simulation until the short chains had diffused their own size, diffused their own radius of gyration. And that's much, much cheaper than our original equilibration. This was days rather than months to calculate. So now we've got a fully relaxed blend that's 50-50, 1,000 and 500 carbons. And we can repeat the process again. We can cut those 500s and turn them into 250s and relax that. Again, that's easy and quick to relax. We can cut again, make another 50-50 blend, and we can branch from here as well. So now, instead of cutting the 250 chains, we cut half of the remaining 1000s. We cut them a couple of times, and we can now create a blend that's 25% long chains, 75% long chains. And these are all quick and easy to equilibrate, which was one of our major simulation costs. And then we've got these nice configurations that we can do our crystallization simulations on. So we did all that, we relaxed them, we then ran the simulations, and we looked at the nuclei as they formed and asked, what's the concentration of short and long chains in the nucleus? So this is the blend. I'm showing you the blend where it worked the nicest. This is polyethylene. It's a 50-50 mix of C1000, our original long chains, with C125. So we've cut these 4 times. So this was the most widely separated blend that we looked at. And we ran our flow simulation. And what we did was at each strain window, so each essentially window in time, we looked at all the nuclei that we detected at that point and asked, what's the composition? We essentially binned them by size, and then each size we averaged the composition across the nuclei that we'd seen in that bin. So this is early on. This is a strain of 2 to 2.5, so fairly early on. And what we see are not many nuclei because we've not really stretched the chains very much. And those that we do are sitting pretty much on the 50-50. So we're seeing a small number of rare, unsuccessful nuclei that correspond to the melt concentration. Then we go a bit further through the flow. Again, not seeing much separation. Further still, we start to see larger nuclei and the separation starts to emerge. So now we're just essentially running through time or strain, if you prefer to think of it that way. And as the calculation goes on, as the simulation goes on, we start to see a systematic separation emerge. So this is the melt concentration. This is the concentration of long chains in the nuclei that we're seeing. This is the concentration of short chains. And as the strain develops, this separation gets wider. And you'll notice that we've introduced hexagons here. So these hexagons correspond to only successful nuclei. So they're nuclei that happen to go on and create stable nuclei, whereas the star ones correspond to all the ones we see. So these are the ones that attempt to grow but then fail. So we see only successful nucleation at the later times in the simulation where the strain has built up. But also what we see is broadly similar separation in the successful and the unsuccessful. If you squint, you might think that the successful ones are perhaps a little bit more separated, but that's likely to be, we didn't see that many successful events, so our statistics aren't quite so strong there. Okay, so we've got clear evidence of long chain enrichment in our nuclei that are coming from our molecular dynamics simulations. And so there's 2 questions we can ask now. Can we quantify the degree of long chain enhancement? Of course we can, because the data that I've shown, we can see that there's something that's quantitative. Sorry about this. So essentially, because these lines are fairly flat, we can say that there's for each strain unit, there's an associated degree of concentration. So then we can plot that, and we plotted it against this order parameter, and our simulations collapsed onto a master curve. And perhaps more excitingly, the master curve that we see is predicted by our model. So our kinetic Monte Carlo model that we solved analytically now predicts this degree of separation. It's essentially precisely what we're seeing in the molecular dynamics simulation. And something really, really interesting happens, which is this is the difference in monomer fraction, the difference of monomers in the nuclei, but we can also count stems as well. We can count essentially chains in the nucleus, which species are those from. And the model says that should also be enhanced, but to a slightly lesser degree. So the slope here is a little bit less. And again, we see exactly that result in our molecular dynamics simulations. So having this model has helped us to ask the right questions of the simulations. Okay, there's another question of, can we also predict the nucleation rate using the same model at the same time? And the answer is yes. So we've got the average time to nucleation here, which is essentially the nucleation rate. Down here, we've got the length of the short chains. So this is our C1000. And as we cut them, we move down the length of short chains. We also change the flow rate as well. So if we flow really fast, we don't see much difference between the short and long chains. But as we slow down in shear rate, nucleation takes longer, but the short chains don't stretch quite as much. And that's where this separation occurs. And also we see and predict a longer nucleation rate, longer nucleation time. Okay, so the final thing that we did in this project was also try to make the models that we've developed more accessible to users. So we developed a piece of software. So this
Richard, I'm just a little bit conscious of time, Richard. So if this is a long section or a...
Okay, I think I can skip this. So we'll go on to the summary. So this is a sort of scientific summary, which really says we use molecular dynamics as a way of complementing experiments, and we embedded that in some multi-scale modeling to help deal with the widespread of time and length scales. And I wanted to get to this slide because this is sort of what did I learn during this project? What were some of the take-home messages for me? Because it was the first time I did a large-scale molecular dynamics project. So my sort of takeaway tips from this project would be use multi-scale modeling to give your molecular dynamics simulations some context and to help you design the projects. Doing these multi-scale projects is hard. Doing the multi-scale modeling and large-scale simulations is a big ask within a single group. So be prepared to collaborate. You know, I certainly wasn't ashamed of not knowing things, and I went and found people that did know it and got them to help me. Think about your software performance. Talk to research software engineers or some other experts on getting the compile optimized to your system. We saw a massive benefit from doing that. Work hard on estimating the compute costs. So in order to get time on the cluster that we wanted, we had to show good estimates of what we were going to achieve, how long that was going to take, how much compute time did we need. And that was really helpful for us in terms of designing the project. And it's actually something that I'm going to be doing now, whether the cluster demands it or not. And my other tip is do apply for time on regional and national clusters if it's justified, because it really helps with your project planning, helps you think about what you're doing, prioritize what you're trying to achieve, and ask what's plausible on the timescales that I'm working with, the timescale of the project, timescale of postdocs, PhD students who need results to finish or get their next job. And so, yeah, that planning I think is a really helpful thing to do and something I'm going to take forward. Okay, I'll finish there. Thanks for your attention. Happy to take questions.
Okay, thanks, Richard. I should say that I've seen quite a lot of this talk before, and one of the reasons that we were keen to invite you is that you see many people give talks where they put up these slides with different length scales and they say, oh, we're going to bridge between them and work all the way up to the macro scale. And nobody ever does, but you did. and then went back and asked questions based on what you learned at the top level all the way back to the molecular scale. So I think it's a really nice example of it can actually be done in a way that produces useful quantitative insights. So thanks very much for coming along and sharing that with the community, Richard.
Thank you.
There's a couple of questions that well, one from me and
Okay.
one that's come in, and then I guess we should try and wrap up fairly quickly because we're over time. So the first question from the attendees was if you could imagine running the MD for long enough that if you had the next but 10 regional cluster and you could run your simulations for essentially infinite time, would you see the polymers fully crystallize, or would you still expect to see some disordered regions due to entropic effects?
Okay, that's a great question. So these are experiments that can be done. So the answer to this question is experimental, in the sense that these are experiments that have been done. The crystals are big enough that you can look at them, and the answer is no. You don't see complete crystallization, because what happens is the chains get stuck. They form these loops. They can't escape the crystal, but the crystal front carries on growing. So the chains find it very hard to reorganize themselves once they're committed to a crystal. The crystal front has moved on. They're essentially locked in. And so you have these dangling ends and loops, and some of those dangling ends go and find other crystals and form those. And it's very, very hard to make a full crystal. And the idea behind this is what you see is not the most stable crystal, but the crystal that grows fastest, which is a different thing. So in some systems, you'll grow the crystal fast and then it will sort itself out afterwards. In polymers, they can't do that. The connectivity stops it from doing that. So yes, if we could run the simulation for long enough, that's what we'd see. Other groups have done this. So groups that aren't so interested in nucleation and more interested in the final structure have run exactly the force field that we used and shown that you get this lamella structure. And then once the lamella structure has grown right the way across the simulation box, it hasn't really got anywhere else to go. The amorphous regions between sit there and stay. And this is actually a really nice feature of real polymers, because the crystallinity gives you stiffness and strength, and the amorphous region gives you a little bit of give. It stops it from being too brittle. So there's a real interest in being able to control the thickness of the lamella compared to the thickness of the crystal compared to the thickness of the amorphous region. And flow is one of the avenues to doing that. It's not something that we had time to get onto, but it's well known in polymer processing.
And there was a software question from me, which is you compared espresso and lamps.
Yeah.
And I guess I'm interested from the point of view of the CCP5 software roadmap. Why were those the two choices for this kind of work?
Okay.
Why is that what you default to?
So it's pragmatic rather than any other drivers. So the postdoc that I recruited to the project had done some early work in this area. So he'd done essentially the same kind of calculations that we'd done, but for much shorter polymers without the interpretation. He'd done the simulations and shown that they were possible using espresso, and he'd modified espresso to include the bits and pieces that he needed. So he was bringing that expertise into the group, and essentially it was one of the reasons I hired him. And so we started off working with espresso, but then we noticed how much work was going into working with lamps. So we tried that out and did a comparison. So it was essentially what was available to us and where existing expertise was helping us.
Yeah, so the expertise within that kind of community in those particular codes.
Exactly. So it was, I mean, what we needed was something that could do flow, something that could incorporate the kind of force field that we need, and it's quite a complicated one. And then having had those two things, we looked for of the options, which ones do we understand, which is the fastest.
Yeah, I guess in particular, flow is not something that's in every package.
It's coming. It's coming. Lamps has it now. In fact, can do extensional flow as well.
Oh.
That's technically much harder, because your simulation box starts as a square and winds up as something very, very long and thin, which is not good.
So I think what we should do is we should probably wrap up formally at this point, since we've ran over time. But I'm also conscious that Richard said he was happy to stay for as long as he needed to at the end of the seminar to answer questions. So I think what I'll do is I'll formally close the seminar for now. But if people do have questions that they would like answered, then maybe Richard would be willing to hang around and answer them via the chat. So we'll stop the recording shortly, and I would